Hello everyone, today I am going to talk about wild eggplant chromosomes and the possibilities of crop improvement. This is a research which is funded by ANRF in 2024. I myself, Dr. Biplab Kumar Bhowmick, the PI of the project, along with me, Dr. Rupsanatan Mandal, who is the co-PI of the project, and Dept, who is working as a JRF in the project. Now, the focus is on eggplants. Why? Because in India, we see this is the native crop which produces almost 27% of the world's total eggplants. There's a huge fruit morphological diversity, like the shapes of fruits and the colors of fruits are commercially important. And along with that also we see some problems in breeding, like we know that eggplants are worst affected by the borers along with fungal pathogens. So what the scientists recommend over time, because since the cultivars are losing resilience over time, scientists are repeatedly telling to introduce genes from the wild stocks. Wild species that are having genetic compatibility, they have resistance to borers, pathogens and abiotic stress, richer in antioxidants and etc. So they can be tested. Now, what wild species should be taken for this purpose? Wild species that are reportedly close to the phylogenetic clade of the eggplant, like Leptostemmonum clade, which bears the eggplants, as well as the wild species which are the closest allies in the primary gene pool. Gene pool 1, that means where the species are interbreeding, and gene pool 2, where the species are interbreeding, the fertility of the hybrids are questionable. So these species can be potentially taken. But the problem is that in India, these species occur in restricted pockets and availability of the taxa becoming really difficult because of habitat depletion. Now what we have taken as an approach, we have requested and collected germplasm from NBPGR, and we are maintaining that and made our center as a conservation center of the wild eggplants. Now what wild eggplants have we chosen? We have chosen Solanum incenum, which is the progenitor of the eggplant, modern eggplant, domesticated eggplant, which is part of the gene pool 1. Then gene pool 2 contains Solanum macrocarpon and Solanum ethiopicum, which are the African giboma and scarlet eggplants respectively. These species contain resistance to fungal pathogens, and that is why they are valuable. Then we have chosen the VD spiny species like Solanum viarum, virginianum and Solanum violaceum, all from NBPGR, having reportedly resistance to borers, and these are also closest allies of eggplant. Now what parameters should be tested before we can go for selection of the taxa for breeding? Morphological characters, phylogenetic and genetic relationships. Both of these parameters are well reported, but chromosome number and karyotype also are inevitable parameters and are not reported in the closest wild gene pools. Why is it important? Because it lets us know the diploid chromosome number, it lets us see the structural differences across the karyotypes, and we can quickly and easily predict whether the chosen wild eggplant is genetically close to the cultivated eggplant or not. So definitely we have the questions in mind whether these will show up similar or different chromosomal makeup, but we have this hypothesis that the progenitor species like Solanum incenum should be very close to the brinjal eggplant because they are the progenitor species, and along with that the other species should also show similarity in terms of the chromosomal makeup. So in order to study chromosome, we should start with pretreatment of the roots. Roots contain the actively dividing meristems. High number of cells are dividing there. So the metaphase chromosomes are easily capturable there. Why? Because if we apply some pretreating and fixing agent, some chemicals which are very well known and easily available, they can be incubated in those reagents and they can enrich the metaphase chromosomes. Why metaphase chromosomes are important? Because metaphase chromosomes show up the condensed morphology based on which we can readily count what is the chromosome number of a species and also see the structural details of that. For this reason, the next step is preparation of the spread of the chromosomes on slides. Conventionally, acetoarsine stain is used to study the chromosomes of plants, and squashing method is used where the cover slip is used for preparing the chromosomes. Now this method has some drawbacks like cell wall remains, and for minute chromosomes like that of eggplants, it is not a very suitable method, but it may work for other materials. For our purpose, we have chosen some enzymes in the alternate method of chromosome preparation. Cellulase spectrulase, these enzymes digest the cell wall and render elimination of the cell wall and cytoplasm, scattering of the chromosomes. All chromosomes lie in one plane. As you can see, this also doesn't need any cover slip. and we can also stain this particular slide with Giemsa and also with the other dyes like fluorescent dyes, and we can see bands on the chromosomes which can better tell apart the species. This is one of the conventional preparations of Solanum melongena chromosomes where chromosomes are almost not visible, and this is one of the enzyme digested route showing up good metaphase chromosomes, and karyotype shows these are the chromosomes having the distinct centromeres, and also the secondary constriction is visible. Now coming to the results, the cultivars have similar chromosome number, 24 chromosomes are there. Chromosomes are mostly metacentric as in the karyotype, as you can see here, Giemsa stained, and this is Dept stained. Dept is a fluorescent dye, and in this we can see the chromosomes better, and we have seen that this cultivated eggplant is very closely allied with the Solanum insenum, which is the progenitor in terms of chromosome structure and chromosome number. Coming to the African scarlet eggplant, the cultivated eggplant of India also has similar chromosome number and chromosome structural features with the cultivated scarlet eggplant of Africa. Now coming to the other VD species, Solanum violaceum and viarum also show almost similar chromosome morphology, and the number is consistent in all of them. So there is a high chances of consistency in chromosome number, as you can see here. Now chromosome size also we have measured in micrometer from the software which is attached to our camera that we use for taking the images of the chromosomes, and based on the statistical parameters we have seen that Solanum insenum and Solanum melongena are the closest, followed by the other species. So coming to the first question, what wild species should this be that should be chosen for introgressive breeding? Definitely we can suggest Solanum insenum, as their literature reports also show this. They are also found to interbreed with Solanum, and by means of chromosomal characters we can also confirm that. Followed by Solanum ethiopium, viarum and violaceum, they can also be potentially tested for introgression breeding. Based on this result, we have got one publication on Solanum insenum and melongena. Rest of the works are underway. We can very soon come up with good and interesting results, not only involving this Giemsa and Dept but also other fluorescence staining. These are few of the seminal references that we have considered for this study, and we highly acknowledge ANRF erstwhile DST-SERB for sanctioning the core research grant CRG 2023-002240 in 2024. We are also grateful to the facilities that are provided by Scottish Church College and the principal of our college for encouraging research activities. Thank you.